Hi everybody, welcome back. I'm hoping you can hear me. I probably have bedhead, but oh well, I guess that's the way it goes. Okay, we are going to continue on now and talk about DNA replication. So again, I love this stuff. This is from the study guide, so I don't know if we'll get through all of this in one screencast or we'll have to do two of them, but I'm just going to work my way through this study guide and you can follow along with your study guide if you want to. All right, so this is a quick review of when in the cell cycle replication is going to happen. So there's a picture down here of the cell cycle, mitosis, interphase, and all this. So DNA is getting replicated during the S part of the cell cycle. That is when all of the DNA in a cell gets copied, is during the S phase. And then at the end of that, remember, we have these two daughter chromosomes that are identical, or they end up being identical to the beginning. Okay, so just a quick reminder to you about where this is happening. Okay, and then we'll get back to Watson and Crick. They were among the people trying to figure out how the structure that they came up with for the DNA could facilitate making copies of it. So this first experiment that I'm going to talk about, this Messelson and Stahl experiment, is coming up. But here are the three ideas that were current at the time when they were figuring this out. Okay, the first model is called the conservative model, DNA replication. And what that suggested was that here's our parent molecule. Both strands are shown in dark blue. And this model suggested that the parent is kept intact. So after the first replication, we have the exact same atoms, basically, and molecules in one copy, and the second copy is just brand new. Okay, so none of the parent is retained in this copy. Then that happens again. So after the second round of replication, this one is copied, and these are all brand new. So that's called conservative because these two original strands are just conserved throughout. The semi-conservative model is the one where the parent strand divides, splits up, and then each parent is retained in one of the copies. So the dark blue one is in this copy, and then a new copy is made, which is shown in light blue. So it's semi-conservative. And then here's the other daughter of that, if you will. One of the strands is the original parent, and the other one is new. So it's partially conserved. It was called semi-conservative. And then that would happen again in the next round. In this first round parent, one strand is retained, and the light blue one is brand new. And then here, the light blue one is retained, but the new one's also light blue. So that's what would happen in the semi-conservative model. And this is actually the model Watson and Crick thought was going to be correct. And then the third one is called the dispersive model, where here's the two parent strands shown in dark blue. And what was proposed here was that those were just sort of randomly, chunks of it were randomly dispersed in the new copies. So both new copies were some from the parent and some that were brand new, sort of spread randomly. Okay, so these are the three models that they were trying to figure out. And this experiment has been called the most beautiful experiment in biology. And I put a link here, which I'll put in the other slides, of a talk. It is so awesome and cute with these two actual guys, they're old men now, talking about their days doing this experiment. It's really fun to watch. It's super elegant because they were trying to figure out these models. So what they did was they took some bacteria and they grew it in growth media that contained heavy nitrogen. So that N15 isotope of nitrogen, the normal isotope is N14. So this one's heavier. And then they could incorporate both isotopes into the DNA as the DNA replicated. And they were going to use this weight difference to figure out, hopefully, which model was correct. Okay, so they grew the bacteria in the heavy nitrogen. And then after one round, they transferred it into normal media containing normal N14 nitrogen. Okay, so that put the lighter isotope into DNA, but only for one round. Okay, and then they spun it. So this centrifuge is going to separate those strands of DNA by mass. The heavier, the one containing the heavier nitrogen will go further down in the tube. The one containing the lighter nitrogen will be not as far down in the tube, so they separate them that way. And then they ran a gel, which, they looked at it, they could analyze the contents of each sample. Okay, so they grew it in heavy nitrogen, they transferred it to light nitrogen, they spun it after one round, and then they grew it some more, and they spun it again. Okay, and this is what they saw. All right, after the first round, if the conservative model were true, you would expect, right, you would expect one heavy band and one lighter band. All right, that was the expectation that they thought that they would see if that conservative model was true. But after the first round, they only saw one band that was intermediate in density. So that instantly ruled out this conservative model. Okay. Then after they still couldn't tell though between these two models, the semi-conservative or the dispersive, because one intermediate band, either one of those could still be true. So then they grew it some more, and after the second and third rounds of replication, they could look at their densities, and this is what happened. Okay, what they observed was two different weighted bands. Okay, if the dispersive model were true, and these two parents are separating and they're replicating dispersively, you would still expect to see a band of one weight. But they didn't. They saw bands of two different weights. So that confirmed that there was a lighter band and a heavier band, and very much confirmed the semi-conservative model of DNA replication. And it was so elegant and simple because in one experiment, they could distinguish between these models. Okay, so this Mendelssohn-Stahl experiment is very famous. It's important to understand it. That talk is really fun if you have time to watch it. Okay, so the semi-conservative model is what we now know. Okay, so let's talk about the process of replication. Okay, we have these things called origin of replications, which is a specific DNA sequence where replication will start. The left-hand side of this figure is a circular chromosome present like in bacteria and E. coli. They have one chromosome and it's circular. And so in this case, this origin of replication will open up and start replicating down in both directions and eventually meeting in the middle. And then it would split. We get two daughter DNA molecules. And that's actually shown in this cool picture down here. Okay, and then in a eukaryotic cell, the chromosomes are linear, not circular. So here's a stretch of double-stranded DNA. And origins of replication will start at all sorts of places along these chromosomes because if we only had one origin of replication, it's going to take a long time. So there's several origins of replication along the length of eukaryotic chromosomes. And we'll get a replication bubble, and then replication will proceed in both directions from those origins, okay? And then they'll just go to get to the end, and then these will merge, and we'll end up with two, complete strands as part of the double-stranded daughter. All right, so it all starts at these origin of replications. This is just a bigger picture, okay? And then that's the beginning, okay? And the whole thing is dependent upon enzymes, which we talked about in Unit 1, okay? Enzymes are generally proteins. They speed up a reaction by lowering the activation energy. So they change the active site of the enzyme and the mechanism so that reactions can go faster, okay? And they generally have, primary, secondary, tertiary structure, etc., okay? And this question, can enzymes alone drive an endergonic reaction? Why or why not? Sorry, I had another coughing attack. Okay, you also need to have free energy in order to drive an endergonic reaction. So enzymes by themselves cannot do it. Okay, but anyway, we're going to talk about 5 different enzymes. Okay, this is just a review basically of what I was just going over. So you can look at that if you need to. That are responsible for DNA replication, and we'll go through these as I talk about the process of replication. But these are the big ones. Okay, helicase, topoisomerase, primase, polymerase, and ligase. Okay, I included single-stranded binding proteins on the slide because they're associated with helicase, but they are not technically an enzyme. Okay, I am going to stop right here and move on to the next screencast.